Okay.
You can start.
Greetings and an excellent evening to everyone present here. I'm Jyotirmayi from Proteus, the Biology Club of IISER, and I shall be your host today. Proteus is delighted to welcome you all to our fourth talk in RHETOR 3.0. Uh, RHETOR started as a mini symposium with guest speakers from worldwide, indulging our audience in various exciting developments in the scientific arena. We are excited to conduct RHETOR with full enthusiasm and at a larger scale this time. The theme of RHETOR 3.0 is etc., a timeline of biological manipulations. We'll explore the concept of the hierarchy of the biological organization from molecular biology to astrobiology and leave it open ended for the future of biology to decide upon the concept. The topic for our fourth talk will be germline protein cup non-cell autonomously limits migratory cell-fate in Drosophila oogenesis. Our speaker for the day is Professor Mohit Prasad. He is a professor at Indian Institute of Science, Education and Research, Kolkata. He is an awardee of Ramalingaswamy Fellowship by the Department of Biotechnology. We cordially welcome you, sir. Uh, Professor Mohit Prasad obtained his BSc degree in botany from Delhi University and MSc in biotechnology from Savitribai Pune University. He obtained his PhD from Center for Cellular and Molecular Biology, Hyderabad. He was a postdoctoral fellow at Johns Hopkins University, USA, where he tried to understand the molecular mechanism of cell migration that has potential implications in cancer metastasis and inflammation. Before we begin the talk, I request the audience to kindly keep microphones muted and video switched off throughout the session. I request everyone to please hold on to your questions until the end of the talk and let us have a fruitful interactive session towards the end. I'd like to hand over the virtual mic to Professor Mohit Prasad now. Thank you.
Thank you. Was it Jyotirmay? Sorry, I missed. Is it Jyotirmay or
Yes, sir.
hello? Yeah. Thank you, Jyotirmay, and thank you, Proteus, for inviting me to this platform. And I thank the biology group of IISER Trivandrum for organizing this symposium. And it is a very nice collection of talks where I see really from molecules, cells, and at the level of astrobiology too, because I see a talk by Devapriya Chatwade, who was our former colleague here in IISER Kolkata. So with this, let me kind of share my slides. 1 minute, just 1 minute. Yeah. Okay, I am trying to log in. Somehow my thing is, just give me a moment. Are you able to see my slides or so? 1 minute.
Not yet, sir.
Okay, let me see if the system preferences for more help go to the issues presenting. 1 minute, I think there is a problem with this one. Basically a recording, so my system is not allowing me to log in because it says that the presenting thing is a problem here. Just give me a moment. System for more help go to the issues presenting. Click on. Yes, I'm trying to troubleshoot this. Just give me a moment. Yeah. System manager, click on security and privacy. Yeah, can you see my slide here, students?
Yes, sir, it's visible.
It's visible? Yeah, yeah. I had to change some settings. Uh, Sorry for this. But I'm not a computer friendly person, so when this came up, it was taking some time for me to sort out. But thank you, everyone out here, because I believe there is finals of T20 going parallelly, and I had been also following the cricket match. Uh, and though it's our neighboring country which is playing, but it's an interesting match which is happening. Okay. And I was just wondering when India was playing through the initial stages and before the semifinals, I was wondering if India would have been here, the situation would have been different today. Okay, so with that, let me start my talk. And basically, I'm a fly geneticist, and we work. We are trying to understand how metazoan morphogenesis is regulated. So I believe there are students who are from the first year, because I see 21 MS or so. You have this roll numbers which were saying 21. So I believe that is the year number or so when you have joined your BSMS program, or it could be PhD. So most of them are BSMS students, or they are PhD students, or there will be a diversity in the group, is it? So, so what we basically try to understand is how intercellular communication aids in metazoan morphogenesis. So if you go by the word morphogenesis, morphogenesis is a very a broad term which talks about, or which involves all the cellular events which kind of gives the form to the gives adult form to the metazoan organisms or multicellular organisms. So when we talk about morphogenesis, this will involve cell proliferation, changes in cell size, shape, cell movement, and so on. Okay, so in our lab, basically we are focusing in three aspects, where we are trying to understand how group of cells get specified, they migrate, and recently we have started looking at the aspect that how epithelial cells change shape during development. Okay, so in today's talk, I'm going to mostly focus on group cell migration. And so if you look at group cell migration, unlike single cell migration, it's a group of cells which are interconnected with each other and they migrate. Okay, and this kind of migration is very prevalent in during development, and majority of these migration, group cell migrations, happens from epithelial cells during development and disease. So you have so if you talk about, if you see epithelial cell has a very distinct polarity with respect to apical, basal, and lateral membranes. And what happens during as a subset of cells, as they change their epithelial characteristic, basically what will happen in a layer of epithelial cells, a group of cells which are going to migrate, they are going to become different, they are going to lose adhesion with the neighboring cells. Like if you can see here, so these cells which are shaded here in brown or chocolate brown, they have kind of started undergoing transformation at the molecular level, and as a result they lose attachment to the other epithelial cells which are holding them together, and they start migrating. Okay, so this kind of migration, so any migration is associated with any migration which starts from epithelial cell is associated with transformation of these epithelial cells to some kind of a mesenchymal characteristics, and that potentiates their movement. So we are trying to understand how a select group of epithelial cells become migratory during development. Okay, now if we talk about group cell migration, and I believe Tamal was the first person who spoke about collective dynamics in group cell migration. So group cell migration plays a very diverse role during development and diseases. So it is, as I mentioned, it is associated with epithelial to mesenchymal transition, because they arise from these epithelial cells, and this is very prevalent during gastrulation, where as the three germ layers are reorganized, so you have these epithelial cells where a subset of cells are going to lose their epithelial characteristics, and they start migrating inside to kind of occupy the internal space of the developing embryo, which in many cases it could be the mesoderms or the endoderm cells. So it plays an important role. Gastrulation, it regulates, it plays a very important role in morphogenesis, specifically in branching morphogenesis, like where you have these lung epithelia and kidney epithelia where the cells are migrating. And then it is also group cell migration plays a very important role in angiogenesis, where new blood vessels are formed and the cells migrate collectively. In survival, group cell migration plays a very important role in wound healing, because I think Tamal would have been in Tamal stock, you would have been exposed to the epithelial sheet of cells as they are migrating. So the epithelial sheet of cells is a very nice model to mimic conditions of wound healing. So here the cells which migrate, the free ends lose their epithelial characteristics and they start migrating to close the wound. Unfortunately, group cell migration is also associated with tumor metastasis. So if you look at group cell migration, how a group of epithelial cells acquire migratory phase and migrate is a characteristic of both normal development and also disease conditions like tumor cell metastasis. So though we know that migratory cell fate is associated with is associated with diverse phenomena, but we really don't have a very or we would say we have scant understanding of how epithelial cells are transformed to the migratory fate. Okay. So we are using fly oogenesis model, and fly oogenesis model is fly is a very nice model, and I believe you people have been reading since your school days that fly is a genetic model system. It's very tractable. You can manipulate the genome very easily. There are large number of mutations which are available, and there are several genes which are homologous to the humans. Several genes which are present in the flies are also homologous to the humans. Okay. And if you look at the pathway, signaling pathway, there are similarities between the flies and the humans. Okay, so we are looking at a fly oogenesis model. And why oogenesis? Because oogenesis is a very good system. If you look at Drosophila ovaries, here there are range of morphological changes happen during development, and this range of morphological changes involves cell specification, where epithelial cells acquire migratory fate and migrate. Their cells change shape, and there are instances of cell growth, cell proliferation, name it. Okay, so those people who have not or not familiar with this system, so if you look at a fly, the fly abdomen, the schematic here, the fly abdomen has a pair of ovaries, and if you are able to pull out a ovary, it mostly looks like a bunch of grapes which you're seeing here. Okay, and each grape globule kind of indicates a reproductive unit of a fly. Okay, so if you look at this schematic here, what happens is that there are several grape globules or female reproductive unit of flies which are arranged in an ascending manner. That means you have smaller globules on one side and the larger globules on the other side. Okay, so it's basically an assembly line where the egg is undergoing development. They bud off from the stem cells. So if you take out, so these are basically several assembly lines of these egg chambers, and if you pull out each egg chamber, basically this is a string of egg chambers at different stages which are trapped. Okay, so on one side is these stem cells from these egg chambers bud off. Okay, egg chamber is basically a reproductive unit of fluid with the egg chamber buds off and goes through 14 stages of development to form the mature egg. Okay, now during this development, there are two phases. One is the pre-vitellogenic phases, which is mostly proliferative, and then is a post-vitellogenic phase, where what happens is that that is the time all the vitellogen protein and gets accumulated in the developing egg, and then you have also a tremendous increase in the cell sizes and overall shape. Okay, so we are basically focusing on this stage 8 to stage 10, where many of these morphogenetic events happens. Okay, and for today's talk, I'm going to focus on a group of cells which are shaded here in red, which basically are epithelial to start with, and at around stage 8, they undergo epithelial to mesenchymal transition, detach from the anterior cells, and then they migrate in between these cells to reach that oocyte boundary. So typically, if you look at this this egg chamber, there are two population of cells. In the inside, there are 16 germline cells, which we call as 16 germline cells, out of which one is this oocyte on the posterior end, and the remaining 15 cells, which are having these blue dots, large blue dot, are these nurse cells. Because they are called as nurse cells because the job of these cells are to nurse the developing embryo, developing egg, okay, to make it ready post-fertilization, so that all the events post-fertilizations are regulated by the maternal factors. And around these 16 germline cells, you have a layer of epithelial follicle cells which are surrounding it. They are mostly around this, at around stage 8, they are around 750 follicle cells. And the ends of these egg chamber are marked by these specialized follicle cells, which are marked here in green, called the polar cells. Okay, so what happens is that during stage 8, these green polar cells basically are going to send a signal which will activate a signaling in the neighboring follicle cells to undergo epithelial to mesenchymal transition. And these cells, along with these polar cells, will detach from the cluster and in between migrate between the nurse cells to reach the oocyte boundary. Now, you might ask me this question: what is the significance of this migration in development? This migration, as these clusters of cells reaches the oocyte boundary, this is the oocyte, it is going to form a fine channel in the eggshell, which will potentiate the sperm entry during fertilization. So in case this migration is defective, or if these group of cells are not able to form the oocyte boundary at a particular stipulated time, then the micropyle formation doesn't happen properly. As a result, what happens is the micropyle is not formed, the sperm entry doesn't happen, and the egg is going to be sterile. Okay, so if you look at it, I'll just show you a movie of this egg chamber, which is labeled here in green. This is the oocyte here, and these are the nurse cells, and you can see this is a movie where the border cells have already formed. In the center are these, which is unlabeled area, is the polar cells, which basically have sent a signal to the neighboring follicle cells to undergo epithelial to mesenchymal transition, and then are getting detached and migrating to the oocyte boundary. So this is the border cell cluster which is migrating. As they migrate, they send active protrusions at the front, and they reach the oocyte boundary. Okay, so so how are these border cells specified? I told you that during stage 8, these here the polar cells are labeled in red. So the polar cells are going to send a chemokine signal, which will activate JAK-STAT pathway in the neighboring cells. So the cells which are shaded here in green, in response to the cytokine unpaired, will activate JAK-STAT, and the cells which activate a JAK-STAT signaling beyond a certain threshold, those start undergo epithelial to mesenchymal transition to be a part of the border cell cluster. Okay, so we know that the polar cells play a very important role in specifying the neighboring follicle cells to acquire the migratory border cell fate. Okay, so when we started this work, one of my PhD students who had just joined the lab, and she was very curious to understand as to what are the factors which regulate the border cell fate specification. So we knew that there are the polar cells are regulating the chemo, sending out the chemokine, but are there any other factors which can also modulate the border cell fate? Okay, so with that, she took a approach based on a phenotype. So in the fly base, fly community has a very nice online system where all it's a database where all the mutant details are cataloged. Okay, so whatever anyone publishes, it is already systematic and entered into the database. And if you give the search for a phenotype or a gene name, you can get all the information which is desired, what is known till that day. It's known as the fly base. So what she did was she went to the fly base, and we said that as we know that female sterility could arise because of improper border cell migration as such. So what she said, okay, let's look at what are the female sterile lines available in the, which are listed in the fly base. So that way she gave a search, and she identified there were 42 female sterile line, and specifically she was looking at those genes which are kind of to be associated with the germline, okay, or the nurse cells, because till that time when we started, we didn't have any information as to how the germline might modulate the fate specification of the border cells. Okay, so she went in that direction because we know that there is a good degree of communication between the germline and the follicle cells during oogenesis, right early on, and in the early part of stem cells, early stages of pre-vitellogenic phases, and so on. Okay, so she was curious to know if there was anything which was in the germline which could be modulating the border cell fate. Okay, so when she did a search, so there were 42 female sterile lines, and this is a representation. So this is a typical assay for migration. So here the border cells are labeled in blue. Okay, and if this is a stage 10 egg chamber, morphologically you can clearly demarcate the identify the stage 10 egg chamber, and in majority of these stage 10 egg chambers, the border cells under white type conditions reach the oocyte boundary. And if there is any mutant condition which is going to affect the migration of the border cells, which will impede the migration of the border cells, it will get stuck somewhere in between the oocyte boundary and the point from which it detaches. So this is a typical example of a migration defect. Okay, so she got a list of genes, and out of which we identified, so this was the list of genes, out of which she picked up one gene which is CUP, which was hypomorphic allele, and which was homozygous viable. Okay, it showed a phenotype later on during late stages of egg formation. They were sterile, but then they were homozygous, it was sterile, and so it was easier to start with a line which were homozygous, where we could see the we could directly pull out the ovaries from these mutant eggs. Okay, so when she identified this, looked at this mutant, she saw that there was almost like 90% of the egg chambers, stage 10 egg chambers, were showing a migration defect. Okay, so that was interesting because it was, I believe it was just the first or the second line, because it was a hypomorphic allele, because for other genes you need to go through a complicated crossing scheme to generate a mutant in the germline. Here the mutant itself was hypomorphic. That means hypomorphic, it was making the protein activity was slightly suppressed, which kind of was potentiating the early development, but it was showing a phenotype at a later stages. Okay, so that helped us to directly look at the egg chambers without undergoing those complicated genetic process, and we saw that there was a migration defect. Okay, now when we went back and checked in the literature what is CUP, so the CUP has been, this mutation was picked up as a part of the EMS screen, ethyl methyl sulfonate screen by Shkubak, Trudy Shkubak, and reported in 1991. Okay, and what it has been reported that this is basically affects the chorion formation. So these chorion layers, the follicle cells are not properly developed, they don't migrate at the appropriate place during egg formation, and the chorion doesn't form. Okay, egg shell is not formed. As a result, the eggs are sterile. Okay, molecularly, this particular gene basically codes for an RNA binding protein. And if you look at this RNA cup protein, it has been known to be, it acts as a translation repressor of various maternal mRNAs, including nanos, Oscar, and so on. So basically, it's it's a translation inhibitor, okay, in sitting in the germline. And also there are reports that it is also involved in the transport and localization of mRNAs in the egg chamber. Okay, so this was something different. So unlike the function, so we are seeing that in this cup mutant, we observe that there is migration defect. So then we wanted to check how this particular gene, which is known to function in the germline, affecting the border cell migration, in which is getting specified from the follicle cells. Okay. So when we looked at, as my student was looking at these egg chambers, cup mutant egg chambers, she saw that the border cells which were exhibiting this migration defect, the size of the cluster was larger. Okay. So when she compared the size of the cluster, she found that in the wild type, compared to the wild type, this cup mutant border cell clusters were almost like 2.5-fold or close to threefold, not threefold, but 2.5-fold or 2.7-fold higher, larger in size. Okay. Now, larger size of cluster could indicate that there's a larger cells which are present in the cluster, or it could be the number of cells in the cluster would be different or would be more. So she looked at the number of cells in the cluster, and what she found that in the cup mutant, the number of cells in the migrating cluster was much more compared to that found in the wild type. So in the wild type, so these are average numbers. So basically, in the wild type cluster, the number of border cells can range from 6 to 10. Okay, and that's the reason we might get intermediate numbers. So that's the reason in a population, when you do average, you get something like 6.4 or so. Okay, but you won't get a chamber at any time which will have 6.4 cells. A 0.4 cell is a misleading kind of thing, but it's just a representation of the population, because some might have even, some might have odd number of border cells. Okay, so the first number is more important out here. So if you look at the difference, so the cup mutant border cell clusters were larger. There were two cells more in an average in the migrating cluster as compared to the control. Okay, so that suggests that probably in the cup mutant, more number of follicle cells are acquiring the migratory trait, which in the wild type it's restricted to 6. Okay, so we looked at given that these border cells are getting specified from the anterior follicle cells, and germline and the cup protein is known to function in the germline, we again cross-check what is the expression pattern of cup. And as reported in the literature, we found that the cup is, so you can see here, this blue staining is the DAPI staining, so border cells are going to get specified from here, and the cup protein is present in the germline. Okay, in the germline nerve cells. Okay, and in the cup mutant, this protein is not. You cannot identify the staining of, or it's indistinguishable. So you can't see any cup protein in the cup mutant egg chamber. So in this background, more number of follicle cells acquire the migratory phase. Okay, so this again suggests that the cup, in fact, is in the germline, and it is somehow regulating the border cell fate from the follicle cells. Okay, but this doesn't become because when you submit a paper with this or with these findings, no one is going to accept this expression pattern as a data to say that the cup is indeed working in the germline to regulate the border cell fate for the follicle cells. So we needed to do an experiment where we deplete cup protein only in the germline and see if that can result in excessive border cell fate from the follicle cells. Okay, so as I told you, initially we had a cup mutant egg chamber which was hypomorphically. So in this egg chamber, you have both the germline and the follicle cells which are mutant. Okay, so you really don't know whether the cup in the germline or the follicle cells or both is helping to limit the border cells limit the number of follicle cells requiring the water sulfate. Okay. So we went ahead and based on the comments from the reviewers, where we were specifically asked to down-regulate cup function in the germline. Okay, and the reason this was difficult, probably at the end I can tell you that cup functions at if during oogenesis, cup has a very predominant function during the early stages. Okay, so using any genetic tools to deplete cup function early on, where it was very difficult because it would stall the oogenesis very early on. Okay, so but somehow we one of my student Sayan, he played around with the conditions with using different by using a trick which allowed cup RNA expression only during the mid oogenesis, and he had to try a couple of temperature shift conditions where the RNA was only expressed during the mid oogenesis, so that the early part cup is there, it's able to push through the early part of development, and the cup RNA aids in downregulating cup function and depleting cup function during mid oogenesis, right at the time when the border cells are getting specified. So when he did that, when he downregulated cup function in the germline, so this is a wild type and here is the wild type border cell cluster. Okay, and when he depleted the cup function in the germline, specifically in the germline, he saw a larger number of border cells. Okay, so what we can see here, that you see that depletion of cup in the germline results in larger number of border cells compared to the wild, compared to the control, supporting the fact that cup in the germline, based on the expression that cup is in the germline, and probably it's regulating the border cell fate from the follicle cells. Okay. To further support this data, we went back and we again we did this experiment in the other way. So we have a cup mutant where you have cup is absent here and cup is also absent in the follicle cells. So in a cup hypomorphic allele, you have cup is mutant in the follicle cells and also in the germline. And in this condition, if cup is functioning in the germline, if we put back cup in the germline, are we able to rescue the limit the border cell fate to the wild type numbers? And indeed it was the case that if you, so if we, I show you the numbers. So in the cup mutant, you are getting around 7 number of border cells in the migrating cluster, and when you where we were able to overexpress cup cDNA in the germline, we were able to restrict it to the wild type numbers, supporting the RNAi data, expression pattern, and so on. So this conclusively sets up that germline function of cup non-cell autonomously modulates border cell fate from the follicle cells. So when I say non-cell autonomously, that means the cup is functioning in the germline nerve cells and it is acting somehow to limit the border cell fate from the follicle cells which are next to the germline neck cells. Okay. So the next question was, how does cup affect the border cell fate? Okay. So I have told you that there is this unpaired cytokine which is which is made by the polar cells, and this is secreted from the polar cells, which binds and activates the JAK-STAT in the neighboring follicle cells. Okay, so you have the binding of this unpaired results in dimerization of JAK receptor, which results in results in STAT activation and dimerization, and STAT goes into the nucleus and activates the genes, including one of the genes which is SLOBO, which is required for the border cell fate specification. Okay, so since JAK-STAT is known to be implicated in border cell fate specification, so and we were seeing larger number of follicle cells acquiring border cell fate in the cup mutants, we wanted to compare the status of STAT activity in the wild type. We wanted to compare the STAT activity between the control and the cup mutants. Okay, and as per our expectation, so here we have done the STAT antibody staining. So here the STAT is localized to the nucleus. Okay, so the nucleus STAT is kind of representing the active STAT out here, and you can see that when we compared the STAT between the wild type and cup, you can see the domain of STAT activation was much broader in the cup mutant compared to the wild type. Okay, and my student kind of analyzed the STAT intensity. The STAT intensity was higher in the cup mutants, and also the number of cells which were showing STAT activation beyond a certain threshold was higher in the cup mutants as compared to the wild type, and this is also being depicted here. Okay, suggesting that indeed in the cup mutants we see higher amounts of, so the cup is functioning in the germline, and when cup is not is mutant, somehow large number of cells on the anterior follicle cells are activating STAT, which kind of is resulting in probably causing excess of water sulfate. But to check indeed it was the case. Okay, so what we did was, in the cup mutant we got a allele of cup. Okay, so if indeed elevated STAT is indeed the cause of the excessive water sulfate in the cup mutant, so what we did was, in the cup mutant we reduced the levels of STAT. So we did this in a condition where this is a cup mutant where there is excessive water sulfate. You can see here. Okay, there's almost like close to 7 number of cells in the cup mutant egg chambers, and when we reduced, we got a allele of STAT where 50, that means 50%, one allele of STAT, that means 50% activity of STAT is compromised. Okay, and what you see that as we get a STAT allele in the background where 50% STAT activity is compromised, or gene is compromised, we see that the numbers get reduced to the wild type numbers, supporting that fact that STAT is indeed. So we have seen that STAT was elevated in the cup mutants, and the rescue of the wild type numbers of border cells in STAT mutant indicates that STAT is indeed responsible for the excessive number of border cells we see in the cup mutants. So after having established that cup is indeed functioning through STAT in the follicle cells to limit the border cell fate, the next question was to how is it doing that? Because if you look at it, cup is present in the germline cells here. Okay, and somehow this green region is there, the STAT is getting activated. So cup function is in the germline cells and the STAT is being limited out in the follicle cells. So how is it that the cup, which is functioning in the germline, can limit the STAT expression in the anterior side? Okay, so one possibility could be, okay, and so this is in the cup mutant, you see that the domain of STAT is getting extended. Okay, so one possibility could be that you have these polar cells, and these polar cells are secreting the ligand unpaired. Okay, so one possibility could be that in absence of cup, it could be that these polar cells source of the signal might increase. So one could be that you might see more polar cells getting specified, or the activity or the amount of that signal from the polar cells might be more. They might be synthesizing more of the unpaired ligand. So first thing to rule out this was that we first we check whether the number of polar cells, generally each egg chamber has a pair of polar cells on the anterior and posterior side. Okay, so first we looked at the number of polar cells. So we got a reporter where the unpaired regulatory sequence is placed upstream of the beta galactosidase. Okay, and this becomes a very nice reporter both for the number of polar cells and the levels of the laxity transport and the levels of transcription from the unpaired locus. So what we did was we looked at the number of polar cells, and if we see the number of polar cells, there are 2 polar cells in the wild type, and same thing we see in the cup mutant. So that means the number of polar cells are like that of wild type, and we compared the levels of beta galactosidase between the wild type and cup. We didn't see any difference between the intensity between beta galactoside between the wild type and cup, suggesting that probably the ligand production is not getting affected in the cup mutants. Okay, so this left us with this led us to ponder further that how cup might be elevating JAK-STAT from the follicle cells. Okay, so we have ruled out the possibility that the ligand production would be affected. So next question was that we started looking at the inhibitors, because one possibility that if cup is not there, cup might be activating some inhibitors in the follicle cells, which would be blocking the activation of JAK-STAT. So when we were looking at all the possibility, all the negative inhibitors of JAK-STAT, Brahma, PTP, SOX, and Notch, Notch was the one which came to our attention. And the reason Notch came to our attention is that Notch is a receptor which is a juxtacrine, which Notch signaling is juxtacrine. So the receptor is present in the follicle cells, and it's well known that its ligand is expressed in the nurse cells. Okay, and during oogenesis, there are several instances, Notch and germline delta is the ligand, they interact to regulate even the number of polar cells and some of the fates of these follicle cells during early part of development. Okay, and given that cup is a germline protein, so could it be that the cup might be regulating delta, and that would be regulating the Notch in the follicle cells, and Notch is one of the negative regulators of JAK-STAT pathway. Okay, so so this is what, so there you have this, you could have the germline delta, which would be regulating the Notch activation in the anterior follicle cells, and we know that Notch and JAK-STAT, so if both Notch and JAK-STAT are antagonistic, higher amounts of JAK-STAT will suppress Notch, higher amounts of Notch will suppress JAK-STAT, and this could be modulating. So in the cup mutant, it could be that this balance might be might be getting impaired. So, but before we go with this, so to check this hypothesis that if indeed it was the Notch signaling was getting affected, so we looked at the Notch signaling in the cup mutant. So we used a reporter for Notch, which is a Notch reporter regulatory element, which is fused to the GFP. And when we looked at this, and this is this is a very nice reporter for the Notch activity of Notch signaling. And when we compared the NRE-GFP between the wild type and the cup mutants, we see the NRE Notch reporter is down-regulated in the cup mutant compared to the control. Okay, so suggesting that Notch signaling is down-regulated in the cup mutant chamber. So what we have seen so far, cup is functioning in the germline. In absence of cup, there is more of JAK-STAT signaling in the follicle cells, and in that background, we also see down-regulation of Notch signaling. And we know that Notch and JAK-STAT are antagonistic. Okay, so given that fact that Notch signaling is down-regulated in the cup mutant chamber, so we looked at the canonical Notch signaling per se. So if you look at the canonical Notch signaling, what happens is that you have a signal sending cell which is presenting the ligand, and you have the signal receiving cell which is presenting the receptor. Okay, so the ligand delta interactions, the ligand receptor interaction for the Notch stimulates stimulates a couple of proteolytic activities, which results in cleavage of the Notch receptor, where there are two components which are generated. One is an intracellular component of Notch, which kind of gets localized, which goes into the cytoplasm and enters the nucleus to activate the Notch target genes. Whereas the Notch extracellular domain of Notch is internalized along with the delta, along with the ligand in the signal sending cells. Okay, so we started looking at what is the status. So we have seen the Notch signaling is down. So we wanted to find out where exactly the Notch signaling was defective in the cup mutants. Okay, so we looked at the levels of NICD and NECD and compared their levels between the wild type and the cup mutant chambers. Okay, so if you see here, this is the top panel is the NICD, and the bottom panel is the NECD. Okay, so in the NICD, you can see the Notch. NICD is, you can see very nice puncta of NICD in the follicle cells. That means the signal receiving cells. Whereas in the cup mutants, we see some punctas, but definitely the number of punctas is much less as compared to what we see in the control. Okay, so this is something. So we see almost 60% of NICD punctas in the cup mutants. Whereas when we looked at the NECD, NECD also was similar. It's just that what we saw in NECD in the wild type, the nerve cells had these endocytosed cleaved extracellular notch receptor, whereas this was not observed, conspicuously observed in the cup mutants. Okay, so what it says that overall, it says that probably the proteolysis of notch receptors is impaired in the cup mutants, because we are not able to distinguish. We see lower levels of NICD in the cup mutants and and lower levels of NECD in the cup mutants. Somehow it looks like the notch proteolysis is not happening in the right way as it is happening in the wild type. So given that notch signaling is down and the notch proteolysis is getting affected, so next we wanted to we asked this question, that is indeed lower amounts of notch signaling which see in the border cells responsible for the excessive border cell fate we have seen in the cup mutant egg chambers. So what we know, that in the cup mutants we have a larger flank of anterior follicle cells activating the JAK-STAT pathway. So our hypothesis was, and if in case, and we in this background we see that the notch signaling is down in the anterior follicle cells. So our hypothesis was that in a background, if we are able to activate notch in the anterior follicle cells, can we reduce or limit the border cell numbers to the wild type? And indeed that was the case, because when we did this experiment where we activated notch only in the anterior follicle cells in the cup mutants, you can see that in the cup mutants you had almost like, in this background we had almost like 8 border cells in the migrating cluster on an average. But when we were able to decrease the overexpression of NICD, was able to reduce the number of follicle cells acquiring the border cell fate in the cup mutant, supporting the fact that notch functions functions downstream of cup to modulate border cell fate. So what we have so far seen is cup is in the germline, notch is in the follicle cells, and cup is doing something in the germline which is affecting the notch activity in the follicle cells. And then once in the cup mutants, the notch activity is lower. As a result, what is happening is that you have excessive border cell fate because of JAK-STAT activation. Okay, so so as I told you that you have notch is activated by as in oogenesis, early part of oogenesis, we know that notch is activated by the ligand delta from the germline. There are several instances there. So we were curious to look at the delta protein per se. Okay, and parallelly what we saw that in the cup mutant, so if you look at the morphology of the cup mutant morphology is very disrupted. So if you see, this is a wild type nerve cell nuclei, here the cup mutant nuclei are very disorganized, and this is a phenotype which is reminiscent where the cytoskeletal structure is disrupted. Okay, and so with this prompted us to look at actin and tubulin, and indeed compared to, we can see very nice framework of F-actin in the wild type, but this is completely absent in the cup mutant. And also nice framework of tubulin, which was kind of not so clear in the cup mutants. So it looks like in the cup mutant, the cytoskeletal in the nerve cells was affected. Okay, so and we know that delta is the ligand which functions from the nerve cells. So we looked at the levels of delta. So when we looked at did a delta antibody staining to see if delta was downregulated, we saw that if you look at the delta, so this is the cup mutant and this is the delta wild type, overall you don't see any stark difference or conspicuous. You don't see a very obvious difference at the levels of cup delta protein between the wild type and the cup mutant. But when you look at it closely, you look at some of the features. So something here. So we know that delta is localized to the oocyte, which is completely, which is downregulated, which is not present in the cup mutant. So that means definitely there is a phenotype. But in the nerve cells, if you look at it, though we see the punctas, when we saw larger number of punctas of delta in the nerve cell of cup mutant chambers. Okay. So we looked at the number of delta particles between the wild type and cup. Overall, there were not differences, but when we counted the number of these punctas out here using automated software, we found there were larger number of cytoplasmic delta in the cup mutant compared to the wild type. Okay. But when we were looking at these two things, we saw there was a difference at something at this level, at here, which you see my cursor. So here it seems to be a more continuous line, and here it seems to be not a continuous line. And it is this place where the border cells are specified from the anterior side. So we zoomed into that part, and then we looked at quantified the delta. So on the face, on the junction between the follicle cells and the nerve cells. So in the wild type, you can see a nice elevation, a large number of delta puncta decorating the interface between the follicle cells and the nerve cells, which was much lower in the cup mutant. So okay, so this kind of suggests that probably there could be in the cup mutant, the delta trafficking might be affected, because we saw there were larger number of delta punctas in the cytoplasm out here in the cup mutants, and here we see that in the anterior side, the delta punctas on this junction was getting affected. Okay, so probably the trafficking of delta is getting affected. Now, if it is a trafficking of delta, when we talk about trafficking could be either internalization of the ligand or the exocytosis of the ligand, because delta is a ligand which is presented on the membrane. So if the trafficking was affected, and so within the trafficking component, we wanted to check whether it's endocytosis or exocytosis which is impaired in the cup mutants. So in this experiment, what we did was we basically took the chambers and we took advantage of the fact that delta if you look at delta ligand, it has a extracellular portion which is presented outside. So if you expose the live tissue with the antibody, so what will happen is this antibody will go and bind to the extracellular tissue, and if it is internalized, okay, so it will be carried further and it will be, and then you fix it. Now, our premise was that in case if it's a defect in the endocytosis, then this delta which is present on the membrane will bind to the ligand and it will be internalized and it will be trapped inside something which we see out here after fixation. And if it is a part of exocytosis, then we won't see any differences. We won't be able to trap these things. The antibody won't get access to the delta protein which is getting exocytosed. Okay, so it has to go to the membrane and then it is going to be internalized. So it will never get a chance to see the antibody because that is happening in the live cell. So the antibody won't go into the cell per se. Okay, so when we did this experiment, we saw that the delta ligand was so we saw that we saw a large number of delta puncta in the cytoplasm, and which suggested that probably the endocytosis of delta ligand is affected in the cup mutants. Okay, now given that endocytosis of delta ligand is affected in the cup mutant, and it is and going by the literature, there are a lot of debates whether what is the role of endocytosis components in mid-oogenesis. So we went further and we wanted to check which segment of endocytosis is impaired. We have seen that delta endocytosis is affected, and if you look endocytosis per se, endocytosis is where the cargo is internalized. It goes through the early endosome, through the RAB5 GTPases, so and late endosome, and to the lysosome, or from early endosome, it might be recycled back to the recycling endosome and presented on the membrane. Okay, and each step is being catalyzed by these RAB GTPases. Okay, so we wanted to check which segment of endocytosis is impaired in the cup mutant, and the idea was that we would overexpress all these RABs in the cup mutant and see which one is going to rescue the border cell fate in the developing eggs. Okay, so what we did was we overexpressed the RABs in the germline cell. So here we are overexpressing the RABs in the germline cell, and we are assaying for the border cells which are getting specified from the anterior follicle cell. Remember that in cup mutant we have large number of border cells, and the idea was that which of the RAB overexpression constructs give you numbers which restrict the number of border cells in the migrating cluster. Okay, so to cut this long set of results, what you can see that when we overexpressed RAB11CA constitutively active, we were in the germline, it was able to limit the border cell numbers to almost the wild type numbers. Okay, we also got some rescue when we overexpress the RAB11 wild type. So compared to the constitutively active, which is even the wild type overexpression gives some kind of a rescue, but the constitutively active was giving a stronger rescue, suggesting that probably the RAB activity is playing a dominant role in is playing a dominant role out here. Okay, so overexpression of RABs here rescues the excessive border cells in the cup mutant. Okay. So as we saw the rescue, the question was, is it specific? And indeed, we looked at the NREGFP notch reporter. When we overexpressed the RAB11CA, you can see that the NREGFP, which was down in the cup mutants, was getting rescued. Okay, and even the RAB delta puncta, which was lower in the follicle cell nerve cell interface, was also getting rescued in the RAB11CA, okay, suggesting that the rescue is specific. Parallelly, when you go ahead with the submission, then the question always comes that, okay, you have done this, you have overexpressed RAB, but this is an overexpression cluster. Can you remove RAB11 function and show that border cell fate is also getting affected? And indeed was this case when we removed the overexpressed RAB11 dominant negative in the germline cells, we got larger border cell clusters. Okay, even the delta punctas were higher in the germline, in the RAB11 kind of reminiscent of the cup mutants. Okay, so which again goes on to say that RAB11 is playing a role in the germline to modulate the border cell fate from the anterior follicle cells. Okay. So last, we wanted to check what is the, because given that we have seen that RAB activity is important, so what is the status of RAB11 protein? Okay, so we looked at RAB11 protein. This is a protein trap where this you have a GFP which is sitting in the intron, which gets spliced and which becomes a part of the translated product. And when we looked at the RAB11, you can see this is the wild type RAB chamber, and this is the cup mutant. And you can see we saw there were a lot of punctas, RAB11 punctas in the perinuclear region, which is becoming very clear. You can see here, it is very clear out here in the perinuclear region of the cup mutants, but not so obvious in the wild type. Okay, so some of the RAB11 GTPs is enriched in the perinuclear region of the cup mutants. So with this, we know that RAB11 activity can regulate its differential localization within the cell. Okay, and Golgi serves as a site where the nucleotide exchange facilitates RAB11 activation. Okay, so we wanted to check whether, and we were seeing that we were getting a very good rescue with RAB11C. So somehow we had this feeling that could it be the RAB11 activity getting affected? And we know that RAB is activated in the trans-Golgi network. So we stained these egg chambers with a marker for the trans-Golgi network, which is Golgi 245, and also looked at RAB11 in the background. So if you see in these panels here, what I would like you to focus is on this last panel here. You can see there are several yellow punctas. This is the nucleus out here. Several yellow punctas where there is co-localization of Golgi 245, the trans-Golgi network, and RAB11 YFP, which is not obvious in the wild type. Okay, so putting these things together, so it says that the RAB11 RAB11 is kind of enriched in the trans-Golgi network in the cup mutant. And given that we see a better rescue with RAB11CA, we believe that in cup mutant background, somehow the RAB11 activity is getting compromised. And as RAB11 activity is getting compromised, delta internalization is not happening, which is required for notch activation in the follicle cells. And in absence, in a condition when notch signaling is down, the JAK-STAT is getting activated beyond the threshold limit, which causes excessive follicle cells to acquire migratory fate. So what are the conclusions of this study? So as I told you, that in the cup, we believe that cup is regulating RAB11 activation, which is kind of potentiating delta endosome recycling, which kind of regulates notch activation in the follicle cells to modulate JAK-STAT activity and kind of limit the number of follicle cells which acquire water sulfate. Okay, and in cup mutants, RAB11 activity is compromised, delta recycling doesn't happen, there's notch is not getting activated. As a result, there is more excessive JAK-STAT signaling and more number of follicle cells acquiring the water sulfate. So with this, and whenever we do this work, so what are the implications of this work? So one thing I would say that this work was possible because of this. Hello, I'm almost done. Hello. Yes,
Sir, continue, please.
yeah, I'm almost done. Yeah. So this work was possible because if you look at, I told you these hypomorphic alleles, somehow hypomorphic alleles are, this work was possible because of the hypomorphic allele. Because as I told you, cup functions has different functions during oogenesis. And presence of this hypomorphic allele allowed us to explore temporally the role of cup during mid-oogenesis. Because if anyone who had taken the cup or mutants, they were stuck in the early oogenesis part. Okay, and all other approaches of RNAi and so on was difficult to manipulate, because you will do a standard RNAi experiment, so you'll say it will get stuck in the early oogenesis. But then once we got a result with hypomorphic allele, we started working on the conditions where you could downregulate cup in the mid-oogenesis. So that is very important. When this work highlights the importance of hypomorphic allele to really dissect, to understand temporal activity or function of a particular gene. Okay. Now, this work also gives important insight into the regulation of notch signaling. Okay. And when I say implications of notch signaling is that but there has been in the field of notch signaling, there is a debate as to how the notch is getting activated once the receptor binds to the delta. So one model is that the pulling force which happens in the signal sending cell once the delta and notch receptor interact, that activates a kind of that activates a protease which causes cleavage of the extracellular domain, and kind of that potentiates the notch activation. There is another model which suggests that delta is recycled, and somehow this recycling of delta is presented on the membrane, which is kind of important for its binding to the notch receptor. Okay, and our data supports the fact that the recycling part of delta is important for notch activation. So somehow we see that our results suggest that the recycling of delta in the germline is crucial for notch activation. Okay, so with this, I would like to thank all the people who were associated with the work. The work was started by Manashika here. She was the person who really started the work, which she started looking at the hypomorphic alleles of the germline and so on. This work was the endocytosis assays, where we looked at delta in the real-time conditions, where we were able to distinguish whether it's endo or exocytosis, or which was impaired in delta. In cup mutants, it was Purbasa here. And Cyan has been instrumental in really determining RAB11, the role of RAB11, and how RAB11 is critical downstream of cup. And Gaurav is also participating at the level of seeing how notch activation in the follicle cells were able to rescue the cup mutant phenotype. With that, I would like to thank all the people who gave us the reagents, and Bloomington Stock Center, DSHB, CCAMP Bangalore for generating the transient line for US Cup. And with this, I would like to thank the anonymous reviewers of Review Commons for critical comments. Still, we are hoping to submit the revised manuscript. I don't know where this manuscript will land, but definitely their comments were helpful in making this thing much more, rendering this manuscript much more clear with respect to what message we were trying to communicate. With that, I thank you, and thank you for your patience. And I'm sorry that I kept you far from this cricket match. I don't know what is the status right now, but thank you so much for being here and listening to our work. Thank you. I'm open to questions now.
Thank you, sir. That was an insightful and delightful talk indeed. Now, I would like to invite any questions from the audience.
So England has won. Okay, good. I just see the score now. So no questions?
I think there are no more questions.
That's fine.
I'd like to thank everyone who attended this talk and got actively involved in the interactive session. I extend my gratitude to our faculty advisor, Dr. Sadananda Singh, and the School of Biology for their continued support towards Proteus. Let's meet on 16th November for an exciting talk by Dr. Satish Kurana. Thank you. Thank you so much, sir.
Thank you, everyone. Thank you so much. Thank you.
Thank you, sir.
Yeah, thank you.